SIGMADAF designs and manufactures membrane bioreactors for the treatment and reuse of wastewater. Our MBR systems are delivered in a self-contained plug-and-play unit and ready to operate. Wastewater enters the equipment after being previously screened and homogenized. In the first chamber, also known as the anoxic zone, aeration-free mixing occurs through an agitator, leading to nitrogen removal in the absence of oxygen. This anoxic zone receives a feed from the mixed liquor inside the second chamber, where the MBR reactor is placed through a recirculation pump located in the control room. Subsequently, water moves from the anoxic zone to the MBR reactor through communicating vessels, being aerated by a blower and fine bubble diffusers and then filtered through submerged membranes. At the bottom of the membranes, another bottom-to-top aeration occurs as a preventive cleaning step and is part of the filtration process. The MBR reactor degrades organic matter, COD, BOD5, and suspended solids through biological reactions facilitated by the present biomass and physical reactions facilitated by the submerged membranes. The elements involved in the filtration process include the membranes and the self-priming pump. The latter produces filtered water free of solids, which is suitable for final discharge into a sewer system or natural watercourse. It is worth noting that by simply adding an ultraviolet disinfection process after filtration, water of a higher quality can be obtained, which can be used for sanitary purposes, irrigation, and similar processes. Controlling transmembrane pressure is a critical factor in maintaining good filtration performance. If this pressure exceeds a certain value, a membrane washing cycle is initiated to return to the appropriate working pressure. This cycle is activated semi-automatically. Regarding the sludge produced during the wastewater treatment, there are two pathways. On one hand, there is continuous recirculation from the MBR reactor to the anoxic tank. On the other, to eliminate excess sludge from the system and maintain biological stability, timed purges are performed. The entire process is controlled through an HMI screen installed in the electrical control room, located in an adjacent room to the compact unit.